To the recombinant DNA technology class. Today I'm going to talk about the recombinant DNA technology, which is heart of biotechnology, and also I'm going to talk about what it is, what is its scope, what are its uses, and what are the challenges and issues it is facing. So let us first consider what is biotechnology, and then we will continue. So we all know that it is the technology that utilizes biological systems, living organisms, or parts of this to develop or create different products. And it is not something new because brewing and baking bread are examples of processes that we have done since time immemorial. We also know that since the human beings they started living at one place, they started doing domestication of plants and animals, and they realized that they had to somehow find out which is the best, and as nature also selects the fittest, so they also started artificially selecting and breeding the animals. Also they found that there are some certain seeds, so when the good seeds are sown, it gives good product. So they started doing it, selecting the seeds and breeding the animals. Now they also saw that heredity is there and there are some variations. So it was then found out by the Gregorian Mendelian principle, and also by the discovery of DNA, that is heredity is due to the genetic material, and then this concept of gene came into being. Now, coming to what is gene, specific sequence of nucleotides in DNA or RNA that is located usually on a chromosome, and that is a functional unit of inheritance controlling the transmission and expression of one or more traits by specifying the structure of a particular peptide, polypeptide, and especially a protein, or controlling the functions of other genetic material. Now, then came the central dogma that DNA is transcribed into RNA, or RNA is translated into proteins. Now, the, this is how the recombinant DNA technology is also popularly known as genetic engineering, which means the simple addition, deletion, or manipulation of a single trait in an organism to create a desired change. The first production of recombinant DNA molecules using restriction enzymes occurred in the early 1970s, and it involves joining of DNA from different species and subsequently inserting the hybrid DNA into a host cell, often a bacterium. Now, the two researchers take the credit of this patenting the first time the recombinant DNA technology in 1974, and they are Stanley Cohen of Stanford and Herbert Boyer of UCSF. So we coming to genetic engineering, it is manipulation and alteration of genes. The process involves isolation, transformation, and expression, and there are applications on plants, animals, and on microorganisms. Genetic engineering is artificially copying a piece of DNA from one organism and joining this copy of DNA into the DNA of another organism. Purpose of genetic engineering is that it allows genes from one organism to be inserted into a cell of a different organism of a different species. And the examples are that we can insert human genes into bacterium, human genes into cells from other animals, and also bacterium genes can be inserted into plant cells. So we will see now that genetic engineering means the DNA from different organisms can be combined. The result is recombinant DNA, and also that bacteria can be engineered to produce human proteins, and human genes can be inserted into another other animal's cell. And therefore the concept of transgenic organisms came into being, organisms that are altered by genetic engineering, genetic material changed by other than random natural breeding. Gene transfer is moving a gene from one organism to another. So transgenic means crossing from one place to another. The genic bit means genes, so it means that bit of genes from different living organisms have been bolted together and spliced into another organism to make a new one which does something which is desirable. So this is just to show the process of introducing a gene or formation of the transgenic organism. Example of transgenic organisms are genetically modified organisms and genetically enhanced organisms. Means some organism which will continue to do the same thing but in an enhanced form. It is also known as homologous expression. And genetically modified organism, some organism which is not supposed to do that thing but it is made to do that and into another thing, and that is known as heterologous expression. And for example, we can see that there is the strawberries are pink and there is a blue strawberry. So recombinant DNA technology, the this technology, it has three particular steps: treat foreign DNA and plasmid with restriction enzyme and DNA ligase, introduce the recombinant plasmid into cultured plant cells, regenerate new plants from cultured cells. Tools of recombinant DNA technology are restriction enzymes, vectors, and hosts. And also, the five stages involved in genetic engineering is the isolation, cutting, ligation and insertion, transformation, and expression. So isolation means isolation of a specific gene from donor. For example, humans, the cells are broke open and then the genetic probe is added. A probe is something which is a single stranded DNA or RNA which will go and attach itself to the desired sequence which are complementary to this and reveals position of the gene of interest. Then isolation of plasmid from bacterial cell can be done like this. Then cutting. Restriction enzymes act as molecular scissors and cut DNA at specific sites called restriction sites. The restriction sites, they are cut and they are basically the palindromic sequences. They are from both the sides and therefore when they are cut, they give rise to sticky ends. And these sticky ends are complementary to the ends of the other DNA and therefore they get ligated there. Due to the formation of hydrogen bonding, the ligase, it is ligated there. Then after ligation, it is inserted. The hybrid plasmid is inserted in the host DNA. And then it is allowed to transform. As it multiplies, it is expressed, multiple copies are formed. Bacterial cell reproduces by binary fission and therefore the bacterial cell produces the polypeptides which is coded by the donor DNA. The summary of steps are: cut with restriction enzymes, the formation of sticky ends from the donor DNA, then ligase bonds sticky ends together, then there is a cloned gene which is recombinant DNA and then it is introduced into the bacteria and where it is allowed to express. The expression is getting the organism with the recombinant DNA to produce the desired protein, and when the protein is produced in large amounts, it is isolated and purified. Now coming to the applications of genetic engineering, farming. Gene farming is a technology that scientists use to alter an animal's own DNA and to splice to new DNA called a transgene from another species. In farming, these genetically modified animals are mostly used to make human proteins that have medicinal value, and the proteins encoded by the transgene is secreted into the animal's milk, egg, or blood and then collected and purified. If we go back to the milestone in 1997, Tracy the sheep, the first transgenic animal to produce the recombinant protein drug in her milk, alpha 1 antitrypsin, treatment for emphysema and cystic fibrosis. It was created by PPL Therapeutics and the Roslin Institute. Second is the xenotransplantation. Xeno means foreign, or it is not self, it is foreign. So xenotransplantation is the transplantation of living cells, tissues and organs from one species to another. However, there are ethical issues and issues with rejection, and there are also issues with viral transmission from one species to another. And in biomedical research, a recent approach to xenotransplantation targets pigs as source of animals with the goal of transplanting pig solid organs such as kidneys, hearts and livers into humans, and in some cases external ex vivo pig liver has been used for temporary perfusion for bridging acute liver failure. Now gene therapy, it involves modifying human DNA either to repair it or to replace a faulty gene. The idea of gene therapy is to overcome the effects of a mutation which causes the genetic disease, and cystic fibrosis is the best known disease where gene therapy has been tried. Gene therapy is the collection of methods that allows correlation of gene defects diagnosed in a child or embryo. So the genes are inserted into a person's cell and tissues to treat a disease. Correction of a genetic defect involves delivery of a normal gene into the individual or embryo to take over the function and compensate for the non-functional gene. So therefore the correct DNA is introduced so that the error is rectified, and that it starts producing normal, the normal genes start producing the normal protein, and the disease is get rid of. So to see the milestone, the first gene therapy was given to a 4-year-old girl with adenosine deaminase deficiency, and this is caused due to the deletion of a gene for adenosine deaminase. And in some children, adenosine deaminase deficiency can be cured by bone marrow transplantation and enzyme replacement therapy, but they are not completely curable. And when we study the steps involved, the first step of gene therapy, lymphocytes from the blood of the patients are grown in a culture outside the body, and a functional ADA clone DNA using a retrovector is then introduced into these lymphocytes, which are subsequently returned to the patient. And as these cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocytes. So, if the gene isolated from bone marrow cells producing ADA is introduced into the cell at a very early embryonic stages, it could be a permanent cure. So we can see that by using the genetic engineering, some other diseases that can be treated by the gene therapy are hemophilia, cystic fibrosis, Parkinson's disease. Coming to the diagnostics test, genetic engineering can produce very specific and sensitive diagnostic tests for many diseases using engineered protein. And the new technology is also opening up a novel ways of delivering medicines to specific targets. Recombinant DNA technology is a modern molecular diagnostic technique, and it is done in following steps. A single-stranded DNA RNA tagged with a radio-labeled radioactive molecule called probe is allowed to hybridize to its complementary DNA in the clone of cells. The cells are then detected by autoradiography, and the clone having mutated gene will not appear on the photographic film because the probe will not have complementary with the mutated gene. And also the enzyme-linked immunosorbent assay is based on the principle of antigen antibody interaction. Infection by pathogen can be detected by the presence of antigens, proteins, glycoproteins, or by detecting the antibodies synthesized against the pathogen. So these are the modern molecular diagnostic techniques. And using the modern molecular diagnostic techniques, now the therapy or the medicine which is coming into being is the predictive medicine, wherein we utilizing the insight gained from the processing of patients data using the artificial intelligence, we can predict that what condition they are likely to suffer from. And also this predictiveness also makes the coming of a new medicine which is preventive medicine based on the risk factor identified, helping the patient to take preventative measures. And also the because of the different composition of genes in different individuals, because of the different mutations, and at that molecular level there is recognition of the patients for personalized medicines, which is very effective in the treatment. And also, since we know what are the conditions, we can anticipate, and this information can be given to the patient so that the patient can participate actively in and improve their healthcare. Because when a person is diagnosed that he has a genes for which will eventually lead into cardiovascular diseases or blood pressure, so the person can be told that these are the lifestyle diseases, and the patient should be control of the lifestyle and should be able to prevent this by doing the participation, active participation. So this is the molecular diagnostics, which is now very much coming into being, and it is the fastest growing segment of the broader diagnostics market, and it is expected to grow from $8.5 billion at present to around $20 billion by 2030. And by growth of the molecular diagnostic techniques is driven by the technology development, the growth of the point of care market. Also, there are multiple techniques for testing genetic material, and some of which lend themselves more readily for miniaturization. Initially, molecular diagnostics were carried out using hybridization methods such as fluorescence, in situ hybridization. Now, later, polymerase chain reaction and microarrays offered high throughput and more accurate tests. And also, now we can focus on three categories of technologies and equipment, that is microarrays, PCR, and DNA sequencing. Next is vaccines. Genetically engineered microbes can be used to produce the antigens needed in a safe and controllable way. And the use of genetically modified yeast cell to produce a vaccine against the hepatitis B virus has been a major success story. And if we see the milestones, The hepatitis B virus, the vaccine against this was the first to be produced and accepted. Recombinant hepatitis B based vaccines. And traditionally, hepatitis B vaccine was prepared by purifying HBsAg from plasma of infected individuals. But the drawbacks are that there is a contamination of active hepatitis B virus or other viruses like HIV. HBsAg has been cloned and expressed in a number of recombinant systems. And it was the first recombinant subunit vaccine to be approved of general medical use. Recombinant vaccine vectors, you can see that there is one vector which is virus which is non-pathogenic, and there is other which is pathogenic. So we can, if we can just take the DNA from the pathogen and insert it in the non-pathogenic, and the non-pathogenic one can obtain the protein surface antigens, which can be, this is the known as the recombinant virus, and it can be used for making vaccine. Applications are also in the microorganisms, for example, production of insulin, human insulin, which is known as humulin. Bacteria can make human insulin, which prevented many diabetes from getting an allergic reaction to animal insulin. Recombinant human insulin preparation represented the first commercial healthcare product, and by inserting the cDNA coding for insulin A and B chains into the individual E. coli cells. Human insulin can also be produced by expressing the nucleotide sequence coding for human proinsulin in E. coli. Now coming to plants, in some of the applications of biotechnology in agriculture are the production of pest-resistant plants, which decreases the amount of pesticide used. For example, Bt cotton is created by using some strains of bacterium, Bacillus thuringiensis, and this bacterium produces proteins that kill certain insects such as Lepidopterans and Cleoptrans and Diapterans. And Bacillus thuringiensis forms protein crystals during a particular phase of their growth, and these crystals contain a toxic insecticidal protein. And this protein exists in inactive as protoxin, but once an insect ingests the inactive toxin, it is converted into active form of toxin, and it kills the insect. So therefore, the specific Bt toxin genes were isolated from Bacillus thuringiensis and incorporated into several crop plants as cotton. And most Bt toxins are insect group specific, and the toxin is coded by a gene named Cry. Similarly, there is a pest resistant plants are developed by using the biotechnological processes. Nematode Miladogyne incognita infects the root of the tobacco plants, and which reduces the production of tobacco. RNA interference process is used for cellular defense, and this is by in all eukaryote organisms as a method of cellular defense. So double-stranded RNA binds and prevents translation of mRNA, and which is known as silencing. And the source of this complementary RNA could be from an infection by virus having RNA genomes or mobile genetic elements that replicate via an RNA intermediate. So similarly, we have agrobacterium vectors are used to introduce nematode specific genes into the host plants. It produces both sense and antisense RNA in the host cells, and the two RNAs are complementary to each other and forms a double-stranded RNA that initiates RNAi and hence silences the specific mRNA of the nematode. And therefore, the parasite cannot survive in the transgenic host expressing specific interference RNA. The transgenic plant thus gets itself protected from the virus. This is genetically modified organisms are produced using scientific methods include recombinant DNA technology. You can see that insects that feed on plant will die, so select for insecticidal cells and cells used for plant propagation, and then these are ready. These are some genetically modified organisms: golden rice and Bt cotton, then aqua advantage salmon, and these are the pictures. You can see normal versus GMO. And any living organism that possesses a novel combination of genetic material obtained through the use of modern biotechnology is the GMO. Plant application, you see that golden rice, which is possible solution to vitamin A deficiency, vitamin A in rice. And a gene which produces vitamin was taken from daffodils and put into rice to help prevent blindness. These are some industrial enzymes produced by recombinant DNA technology. You can see that in the table. And now coming to the future scope of RDT, that is extremophiles. Hypothermophiles are the hot spring optimum temperature for growth at or above 80 degrees C. And then there are halophiles. The hypersaline waters can grow in 5 molar sodium chloride solution. Then psychrophiles, extremely cold environment. Alkylophiles, they are alkaline environment, and acidophile can withstand the acidic environment. So when these extremophiles, we can study the genetic makeup of these, and we can use the we can compare their genetic makeup, their sequence with the sequence present in the human beings, or where it is required, and this it can be the slight change can make the normal enzymes, it will convert into the extremophiles, and they can be used in various industries, like whether it is the leather industry, the milk industry, food industry, cheese making, bread making, and every and this can be used. So this is the future scope of RDT. Now, coming to RDT issues and challenges, firstly the ethical issues are there, then the safety issues in recombinant DNA technology include gene pollution, because what is happening is the environment is resulting in the antibiotic resistant probes, health effects of food from genetic modified organisms, and there is some allergenicity and adverse immune reactions, effectiveness of pharmaceutical compounds produced using rDNA technology. So uh, with this note, and with these sharing these issues and challenges, I complete the lecture on recombinant DNA technology. Thank you.